Right. And we are recording. Just checking okay, checking the chat one more time, and here we go. So last Thursday, we talked about how proteins have these extremely complicated shapes, and we can describe the shape on 4 different structural levels. So we talked about primary, secondary, tertiary, quaternary structure, and what proteins do depends on proteins having the right shape. If you make muscle proteins, for example, that don't have exactly the right shape, you could end up with one of the 50 or so different diseases that are collectively classified as muscular dystrophy. If you make hemoglobin, the oxygen-carrying blood protein, with the wrong shape, you could end up with thalassemia or sickle cell anemia or glucose 6-phosphate hydrogenase deficiency, or any one of a number of other conditions that have to do with blood. One moment, we've got visitors. Hi, where do you need to go? Okay, go right ahead. Okay. All right. So making proteins with the right shape is critical. And it's particularly critical when the proteins involved are what we call enzymes. And enzymes are biological catalysts, which simply means that they lower the activation energy of a chemical reaction. Remember that many chemical reactions are like the Rolling Stones, because if you start them up, they'll never stop. That was a song back in the old, if you start me up, if you start me up, I'll never stop. Right. And a lot of chemistry works like that. Reactions will give off more energy than they require. And so once you get them going, they'll continue to run. But to start them, you need an input of energy called the activation energy. Just like, a spark is needed to start a flame. What catalysts do is lower the activation energy so that a chemical reaction can start spontaneously and doesn't need nearly as much of that energetic input in order to get rolling. I've compared it as if you have a ball at the top of a hill. Once that ball gets a push, it'll roll until it gets to the bottom. What a catalyst does is remove the speed bump in front of the ball so that instead of being stuck at the top of the hill, the ball can just spontaneously begin rolling. So that's what enzymes do in living systems. They are biological catalysts. And there's our diagram. Without the enzyme, there is a big bump in front of the ball, so it can't spontaneously roll downhill. The bump is much lower, and it's much more likely that the ball will start rolling in the presence of a catalyst, or in living systems in the presence of an enzyme. An enzyme is just a biological catalyst. The reason why shape matters is that an enzyme only works on one or a limited number of molecules that we call the substrate. And an enzyme molecule will have a single region called the active site, and the substrate fits into the active site like a key in a lock. If an enzyme has an active site that doesn't have the right shape, it won't work. I always think of enzymes as being like, socket wrenches. The handle could be whatever shape, it may not even matter that much, but the actual socket is the active site. And if the socket doesn't have the right shape for the bolt you want to turn, or the nut, or whatever it is, if the socket doesn't have the right shape, then the socket wrench is not going to work. It's similar with enzymes. The substrate fits into the active site, and once it's bound, the enzyme molecule can stress the substrate can pull on it. It may bring it next to another molecule that also fits, and the substrate might either be broken up, or in some cases it might be joined together, because enzymes can put together as well as take apart. Okay, this is all review. This is actually slides from last Thursday. But there's a diagram of more or less how it works. The substrate at the beginning is that green shape. It fits into the active site. The enzyme might actually change its shape a little bit as the substrate fits in. That's called induced fit. And then the enzyme causes the substrate in this diagram to break apart. And so the products fall out once the substrate has broken. But enzymes sometimes work in the other direction and might bring two substrates together and weld them together. You know, like many tools, you can use them to break down or to build up. And again, the best metaphor I can think of is a socket wrench, where the socket's the active site and it can only fit one size of shape of nut or bolt. So you saw this on Thursday. That's a model of alpha amylase. That's your saliva enzyme that breaks down the starch that you eat. So again, if you just put a saltine cracker in your mouth and hold it there, as your saliva starts soaking into the cracker, the amylase starts binding to molecules of starch. Diagrammed in yellow, that's a fragment of a starch molecule that has fit into the active site. And once it's in the active site, the starch will get broken down into simpler sugars. This is why if you put a saltine cracker in your mouth and just let it get moist with your saliva, first of all, it'll start falling apart. And second of all, you'll get more and more of that not powerfully sweet, but kind of slightly sweet, malty flavor that crackers tend to have. That's a direct consequence of the starch in the cracker getting broken down into simpler sugar molecules. And then this was where we ended. That's a model of a bacterial enzyme called beta-lactamase. Here, the beta sheets are shown in red, the alpha helices are in green, and the parts that aren't either are shown in blue. But that thing over on the left, right here, that's a molecule of penicillin. And in 3D, you can see it fitting into the protein's active site right there. So there's the overall protein. And the penicillins fit into the active site. And the reason this matters is that when penicillin fits into the active site, beta-lactamase causes it to break. Bacteria that can make beta-lactamase are resistant to penicillin. Some of them can even eat penicillin and use it as an energy source because they've got the enzymes needed to break it down. So that is the reason why we have more and more cases of antibiotic resistant bacteria. They're resistant because they make the enzymes that can break down the antibiotics that we're trying to kill them with. And that's why there's this arms race between bacteria evolving resistance and drug companies trying to develop new antibiotics. Okay, now this is new. An awful lot of your metabolism depends on multi-step reactions. And you can think of these as being kind of like modern assembly lines, where a substrate might get acted on by one enzyme, and then a second, and then a third, and then a fourth, and then a fifth, and so on, with each enzyme performing one single step in a series of chemical reactions. The example that I'm going to show you involves breaking down ethanol. That is the alcohol present in alcoholic drinks. Not that any of you would know because most of you are under 21, but if you drink ethyl alcohol in large quantity, you start screaming a lot, wearing your underpants on your head, and passing out on the porch of the Pi Kappa Alpha house. At least that's what my college roommate always did. Your body does process ethyl alcohol after you drink it, and your liver cells make an enzyme called alcohol dehydrogenase, and that converts ethanol to a chemical called acetaldehyde. Acetaldehyde still is not real good for you. If it builds up in your body, it's toxic, it causes headaches. In fact, acetaldehyde causes a lot of the symptoms of a hangover. So you have to get rid of acetaldehyde, and a second enzyme called aldehyde oxidase breaks down the acetaldehyde and turns it into acetic acid. Acetic acid you can deal with. Acetic acid is the active ingredient in vinegar. Household vinegar is, I think it's 5% acetic acid, and that's what gives it its taste and its smell. Acetic acid is less toxic, and you can excrete that or you can break it down in other ways, but it's not going to harm you the way that too much ethanol or too much acetaldehyde would. So you have this two-step process in breaking down alcohol. One step turns it to acetaldehyde, and the next step turns it to acetic acid. So that's what an ethanol molecule looks like. That's the active ingredient in tequila, or whatever it is you would drink if you were of age, which most of you aren't. Alcohol dehydrogenase converts that ethanol to a molecule of acetaldehyde, which is, that's what that looks like. Acetaldehyde is still nasty, so a second enzyme that your liver cells make called aldehyde oxidase converts acetaldehyde to acetic acid, and acetic acid you can deal with. Acetic acid can be broken down further or ultimately excreted, but it's not nearly as toxic as ethanol or acetaldehyde is. Here's the fun part. This is a fairly common mushroom called the inky cap, or a Coprinus atramentarius, and I've actually seen this growing around Conway. It tends to grow on old stumps, and it's edible, but there's a chemical in that mushroom that blocks aldehyde oxidase. So if you eat Coprinus atramentarius, what happens is that this step right here gets blocked, and you can no longer convert acetaldehyde into acetic acid. And the effect lasts for 24 to 36 hours after you've eaten the mushroom. The upshot is that if you eat these mushrooms and you drink alcohol, your liver cells will start detoxifying the alcohol, and they'll break it down into acetaldehyde with alcohol dehydrogenase, but then they can't get any further because this chemical in the mushroom has blocked aldehyde oxidase. Acetaldehyde starts building up in the body. The end result is you get hit with a hangover from hell. Acetaldehyde causes a lot of the hangover symptoms, like skin flushing in some people, and especially headache. In other words, you can eat these mushrooms, but you must not serve wine with the meal, at least if you care for your dinner guests at all. So that is a consequence of what can happen when you block one enzyme in a series. And I should mention there's a few other compounds that do that. There's a drug called disulfiram that's sold under the trade name Antabuse, and another one called Tempacil, that are used to treat alcoholism because they do the exact same thing as the mushrooms do. They block aldehyde oxidase. So if you take these drugs and then you drink, the ethanol gets broken down into acetaldehyde, but acetaldehyde builds up because your body can't deal with it, and you get an absolutely rotten hangover before you even start feeling the effects of being drunk. You know, it doesn't really treat the underlying psychological compulsions of alcoholism, but it can be very effective in the short term because if you try to drink, it'll make you sick as a dog. It certainly tends to discourage drinking. So it can be used as part of the process of treating alcoholism. I hope none of you ever have to take that stuff. And as I've said, you can think of metabolism as being like a huge set of assembly lines. This is a modern auto assembly line, and each of those yellow things is a robot. And you have a car body on the conveyor belt. The conveyor belt moves forward. You know, the car body comes up to one robot, and the robot will do exactly one thing. You know, maybe tighten one set of screws. And then the car body moves on down the line to the next robot, and the robot will do one thing, like tighten 3 more screws in a different position. And then the car body will move to a different spot, and the third robot will do something, and so on. Every robot carries out one step and can do it for 24 hours a day, never getting tired. Enzymes work a lot like this. Every enzyme is a robot, is a machine that carries out one step in the assembly. And the assembly line of your entire metabolism, if you diagram it out, it looks like this. And I hope you're getting all this down. I'm kidding. Don't even try to get this all down. But every one of those thin black lines is one chemical reaction that your cells can carry out. And every one of those reactions happens because your cell has an enzyme that can carry it out. Every one of those changes, every one of those chemical reactions exists because there's an enzyme that causes it to happen. Okay, that's metabolism for you. Now, right in here, I think, we'll focus in on that little part of the full metabolism of a typical cell. That part that I circled, the technical name for it is glycolysis. And one important component of your foods are simple sugars, notably a sugar called glucose. Glucose you have probably heard of. If you have too much glucose in your bloodstream and your cells can't figure out what to do with it, that's essentially diabetes. And you use glucose as a very important fuel for a lot of the rest of your metabolism. And in glycolysis, you start with glucose up here, and an enzyme called hexo-6-phosphate converts glucose into glucose 6-phosphate. And then a second enzyme called phosphoglucose isomerase converts glucose into fructose 6-phosphate, and so on. Every one of these names in red is the name of an enzyme that turns one molecule into a different molecule. Over in blue on the left, these reactions have the side effect of creating molecules called NADH and ATP. We don't need to worry about what those do yet. Let's just say that those can transport energy and make it available to other chemical reactions. You extract ATP from the food that you eat, and ATP can contribute energy to other things that you need to do, like moving. or other chemistry that we haven't gotten into. We don't really need to worry about what all this is for just yet. But every one of these names that ends in ACE is an enzyme. So we've got 1, 2, 3, 4. That's a fifth right there. 6, 7, 8, 9, 10 enzymes that start with the simple sugar glucose, and at the end of the assembly line or disassembly line, turn it into a molecule called pyruvate. Who cares? This is a picture taken through a microscope of a single-celled fungus, and you ought to remember that single-celled fungi are called yeast. Specifically, this is a yeast called Saccharomyces cerevisiae. And what the yeast can do, if there isn't any oxygen present, is follow up glycolysis with another set of reactions called fermentation. And so what the yeast does is it ferments pyruvate through several steps, and pyruvate ends up getting turned into ethyl alcohol and carbon dioxide gas. And also the yeast gets some energy that it can use to run other reactions in its metabolism. Why does this matter? Well, if you take a liquid that's rich in sugars, like grape juice, and you add some Saccharomyces cerevisiae, you add some yeast, the yeast will start breaking down the sugar. And once the oxygen's used up, they will switch to fermentation, and they'll convert all of that sugar in the grape juice into ethyl alcohol and carbon dioxide. And lo and behold, you've got wine. If you give it a different fruit juice, you'll have, a different wine. You'll have blackberry wine, or was it Trisha Yearwood who sang this? You'll have strawberry wine, 17. Sorry, that's my obligatory attempt to sing a female vocal line that my voice is really not suited for. I have to do this regularly. Right. If you turned your yeast loose on honey, you'd end up with a drink called mead. What are some other examples? Well, let me get to another example. So that's basically how you make wine. If you want to make a different type of drink, you can start with a grain called barley. That's what barley looks like when it's growing. And barley does not contain an awful lot of sugar. It does, however, have a lot of starch, and a starch is a polymer of simple sugars. Somebody mute, please. And here's the thing. Barley seeds contain starch because when you plant a barley seed, the plant embryo inside is going to break that starch down into sugar and use it as fuel until it can get tall enough to sprout leaves and start making its own food. So the starch inside a barley seed is food for the developing embryo until it grows enough that it can do photosynthesis. So what some genius about 7,000 years ago, probably in what is now Iraq, what some brilliant man figured out back then was that if you soak barley seeds in water, spread them out and let them dry, which is what this guy is doing, every seed will begin the process of sprouting. And as sprouting begins, every seed begins making enzymes. And what the enzymes do is they start breaking down the starch in a barley seed and turning it into simple sugar. And by the way, barley that's just begun to sprout is called malt. This is called the malting process that this guy from the Rogue Ale Brewery is doing. He's malting the barley. And of course, if you malt some barley and then grind it up, you can extract this sweet, syrupy stuff. So if anybody's ever heard of getting a chocolate malt, what that means is a chocolate milkshake that's had an added little bit of malt syrup added for extra flavor. Okay, so starch, again, is a polymer of simple sugars, and the seed begins to produce enzymes called amylase. And the amylase that's produced by the plant embryo in the seed break the starch down into a simple sugar called maltose, which the seed would normally use as its energy source. However, you can take that malted barley, grind it up and soak it in hot water to create this sweet extract with this sugar in it. If you want to get technical, that sweet extract is called a wort. And then you take that wort and you feed it to Saccharomyces cerevisiae, the yeast. The yeast can turn the maltose into glucose. It can break down the glucose into pyruvate, and then it can ferment the pyruvate and end up with alcohol and carbon dioxide. And the end result is beer. Traditionally brewed beer is made in such a way that it not only retains the alcohol, but it also keeps the carbon dioxide from fermentation. And that's what makes beer bubbly. Although most of the beer that you'd actually buy has carbon dioxide pumped into it at the factory to, give it a big head and make it extra fizzy. But at least originally, the bubbles in the beer were there because brewers were able to keep the carbon dioxide gas that the yeast were making, were able to keep it dissolved in the beer. By the way, champagne is made in the same way. With most wines, the carbon dioxide is allowed to bubble out. But with champagne, a French monk by the name of Dom Pérignon, yes, that Dom Pérignon, figured out a way to keep the bubbles in the bottle without the bottle exploding, and invented sparkling wine. And that's true for other sparkling wines as well, like, Asti Spumante or what have you. So there, now you know. By the way, when you add yeast to, never mind that, when you add yeast to bread dough, it does exactly the same thing. When you add yeast to a mix of flour and water and sugar and salt and whatever else you've got in there, the yeast starts breaking down the sugar, turning the sugar, first breaking it down and turning it to pyruvate, and then fermenting the pyruvate to ethyl alcohol and carbon dioxide. The carbon dioxide makes bubbles in the dough, and that causes the bread to rise. That's why yeast bread is, if it's made well, is light and fluffy and has all of those bubbles in it. The bubbles come from the live yeast that was added. There's ethyl alcohol in bread dough as well. And theoretically, you could get drunk by eating a huge amount of raw bread dough. Realistically, you just make yourself sick. Don't actually try it. The reason you don't get drunk from eating bread is that the alcohol evaporates when you bake the bread. Some of you might know that bread that's made without something to make it rise can be very, very dense and not fluffy and more like crackers. An old word for yeast was leaven. So if you've ever heard of unleavened bread, that's bread made without any yeast, and it doesn't rise, and it ends up being a hard, crunchy flatbread type of thing instead of a big, poofy, fluffy loaf of bread. Not that I want to turn this into a baking class, but there are a few other things you can do that will substitute for yeast to put gas bubbles in your dough. That's what baking powder does. So there are some styles of bread, like biscuits, that get light and fluffy but don't require yeast. But certainly for traditional loaf bread, you add yeast and you give the yeast time to break down the sugar in the recipe, and it'll make these gas bubbles. And the same is true if you homebrew beer. I haven't done this in years, but I used to. I've seen it. If you add the yeast, your wort starts getting very, very bubbly. And when you bottle it, if you do it right, if you bottle it at the right time and add just a tiny bit of sugar, the yeast will ferment the sugar enough to make your bread fizzy, not enough that the bottles start exploding. So there, now you know. If you wanted to make stronger drinks, by the way, you could brew a beer for whiskey or a corn beer for bourbon or typical moonshine. You could brew a wine if it's brandy we're talking about. You can make a sort of beer out of grain or potatoes for vodka, and then you distill it to concentrate the alcohol. You heat it and collect the alcohol vapors. And then the distillate you might store in a wood barrel for a long time in order to mellow the flavor out. So not that any of you have ever done this because you're mostly underage, but when you get old enough, you might go down to the Rocktown Distillery in Little Rock and see how this is done. Okay, right. How do we tie this back to genes? British doctor named Archibald Garrett in the early 1900s was studying some rare genetic diseases. And one is called alkaptonuria. And this is kind of weird. If you have alkaptonuria, your urine contains high levels of a chemical called homogentisic acid. And homogentisic acid is transparent, but it reacts with oxygen in the air and it starts turning black. So on the left, that's a urine sample freshly collected from someone with alkaptonuria. B, that's a urine sample from somebody with alkaptonuria that's been sitting for about 30 minutes. So if you have alkaptonuria, your pee turns black once it's exposed to air. It's kind of weird. And it's a little more serious than just that. I mean, if all it was that your pee turned black, it would be an interesting party trick and nothing else. But homogentisic acid, your kidneys aren't able to excrete all of it. And homogentisic acid builds up in the skin. This person's ear is actually showing this kind of blackish coloration because homogentisic acid is building up in it. So if you have alkaptonuria, your skin can turn kind of a gray. It can build up in the eyes and the heart, and especially in the joints where it damages your cartilage, which means that if you have alkaptonuria, you're going to develop severe arthritis, typically by the time you're in your 30s. So this is definitely not something that you want to have. Fortunately, it's rare. And it runs in families. And here's why it happens. One of the 20 amino acids that you need to build proteins is an amino acid called tyrosine. And it's an amino acid because right there is that amino group with a nitrogen and some hydrogens. Right there is the carboxyl group. Remember that we often don't even include carbon atoms because they're so common, but there and there we have two carbons. So that's the amino. There's the carboxyl. And then the R group over here is a big ring of carbons and hydrogens. Now, you do need tyrosine to make proteins, but most people get more than they need. So most people have excess tyrosine, and your body disposes of that. And here's the trick. Tyrosine goes through several steps that I'm not including, And after several steps, each one driven by an enzyme, it's turned into a chemical compound called homogentisic acid. Okay, homogentisic acid is the stuff that builds up in the bodies of people that have alkaptonuria. Homogentisic acid normally gets converted by an enzyme to another compound called 4-mallalacetoacetate, and then that gets broken down by several more steps, and ultimately you can use this as an energy source. You can convert it to a form that you can break down for energy. Every one of those steps, every one of those arrows depends on a different enzyme. But if you're missing an enzyme called HGD, stands for homogentisate dioxygenase, don't worry about the name. And remember, I'm never going to test you on the details of case studies, so I'm not going to ask you detailed questions about alkaptonuria. The only way you'd really need to know that would be if you become a doctor, and even then, many doctors never see this. I'm not teaching you about alkaptonuria. I'm teaching you about the link between all this enzyme stuff and the genetics that we're working on. And if an enzyme called HGD is faulty or it's missing, that pathway is blocked. Tyrosine can be converted into homogentisic acid, but it can't go any farther. You can't convert homogentisic acid into this stuff, the 4-mallalacetoacetate. And homogentisic acid builds up in the body. Your body does its best to shovel out the excess homogentisic acid, so a lot of it gets excreted in your kidneys, and that's the stuff that makes your pee turn black. But your body's not able to excrete all of it, so homogentisic acid gets built up in the skin, which can make parts of your skin turn gray, and it builds up in your cartilage, which gives you severe arthritis at a pretty early age. Where's the link? Garrett studied families that have a history of this, and he did analysis of a pedigree like you're going to do in the last lab, and he worked out that alkaptonuria was inherited as a plain old, simple Mendelian recessive allele. It follows exactly the same rules as white flower color back when we were learning about Mendel's pea plants. So from studying this and studying some other human genetic diseases, there were 4 that he worked on, and he found that all 4 followed the same pattern. And he concluded that what a gene had to be was instructions for making one enzyme. And as he put it, one gene, one enzyme. What a gene is something that tells a cell how to make one enzyme. If you inherit a gene that gives you the wrong instructions, your cells can't make the enzyme that you need to break down homogentisic acid, and you end up with black pea and a bad case of arthritis. I was talking earlier about breaking down ethyl alcohol, right? You drink ethanol, you break down ethanol using one enzyme to acetaldehyde, and then you take that acetaldehyde and use an enzyme called aldehyde oxidase to break that down to acetic acid. There are some people that are just born with alleles for a form of aldehyde oxidase that is less active, and they show alcohol intolerance. They always have a bad reaction to even small amounts of alcohol. On the left is a man before drinking alcohol, and on the right, that's a picture of him 5 minutes after drinking a shot, and you can see his skin is turned red. That's a common symptom of alcohol intolerance. So people like this naturally, cannot drink very much alcohol because it will cause acetaldehyde to build up in their bodies and they get a bad reaction. Okay, so there's people that are, it's as if they have always eaten coprinus mushrooms. So there. This is an enzyme that happens to be called phenylalanine hydroxylase. And right there in the active site is a rather complicated amino acid called phenylalanine. And normally, phenylalanine hydroxylase will take phenylalanine and convert it to the amino acid tyrosine. A person who can't make phenylalanine with the right shape, excess phenylalanine will build up in the body, and the body will try to excrete it, but it can't get it all, and that excess phenylalanine starts getting toxic to the nervous system. That buildup of phenylalanine is called PKU, or phenylketonuria, and if it's not caught early, it will cause developmental delays. It'll damage that developing brain. Fortunately, it's pretty easy to test for this, and every newborn is tested for PKU. It's a very non-invasive test. All they have to do is add a chemical to a wet diaper. So every newborn in a hospital is tested for this on the first day. They don't even have to take blood. And if you look very carefully at the labels on things like Diet Coke, notice the warning label, phenylketonurics. That's people who have phenylketonuria. This contains phenylalanine. If you have PKU, you can live a normal life, but you have to eat a very carefully controlled diet that doesn't have any phenylalanine in it. You have to avoid foods with high levels of phenylalanine. And you might remember that that sweetener, aspartame, aka NutraSweet, is a peptide between two amino acids, one of which is phenylalanine. If you drink a lot of Diet Coke, you're taking in a lot of phenylalanine. Most of us can handle that, but people with PKU, at least in childhood, will suffer damage to their brain if they drink Diet Coke. And that's why we have that warning label. Woof. And PKU is inherited as a simple Mendelian trait, right? If you have two copies of the normal allele for phenylalanine hydroxylase, your body will process phenylalanine as normal. If you have one copy of an allele for normal enzyme and one copy of the allele for defective enzyme, you'll still make enough normal enzyme that, you know, you'll be fine. But if you have two copies of alleles for defective phenylalanine hydroxylase enzymes, your body will not be able to break down phenylalanine and you'll have PKU. And you will have to live on a very carefully controlled diet, at least until you're an adult. After adulthood, the effects are not nearly as bad. So that's Garrett's insight round. Now, a gene is responsible for making one polypeptide chain of amino acids. This applies to enzymes. This also applies to proteins that are not enzymes. The proteins in your muscle that make it contract, for example, aren't enzymes, but they still have to have a particular shape. And they have that shape because there are genes that carry the information needed to make them. The instructions for putting amino acids together in the right order are literally encoded in the order of DNA bases. That is why the order of bases matters. That's why it is important that you have a strand of DNA somewhere in your chromosomes where the order of bases is ATG, AAG, TTT, AAC, CC, CC, AT, 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 CCC, or whatever it is. That's what we call the genetic code. That's the importance of DNA. The order of the bases on the 6 feet of DNA in every one of your diploid cells, the order that the bases are in carries the instructions for putting amino acids together in the right order. If you put amino acids together in the right order, that chain of amino acids will fold into the right shape, and that protein can do what you need it to do. If your DNA instructions tell your cells to put amino acids together in the wrong order, the protein will fold into the wrong shape, and your body won't be able to do what that protein was needed for. So it all comes down to shape. What? There, I'm assuming that you're all thoroughly impressed. Okay, that would actually be a good pause point, but we've still got 20 minutes to go. So I'm going to keep pushing on. DNA is located in eukaryotes anyway. DNA is located in the nucleus. In prokaryotes, it's kind of clumped together in the center, but there's no nucleus in prokaryotes. In eukaryotes, DNA is in the nucleus, and it normally stays there. If DNA is coming out of a eukaryotic cell's nucleus, that cell is probably about to die. That cell has been very badly damaged. And yet, it was known back in the 1950s that proteins are made outside the nucleus. So how does the information in DNA get from the inside of the nucleus to the outside where the proteins are actually assembled? Well, pretty soon after DNA structure was discovered, Francis Crick proposed what he called the adapter hypothesis, and that is that information is copied onto a second molecule, and that second molecule shuttles out of the nucleus. Think of it like this. We've got a library on campus. You might have heard of it. It's got a lot of computers and a Starbucks. And it even has books. I know, hard to realize, right? But libraries actually do contain books. And there are some books that you are not ever allowed to take out of the library. They're over in the reference section. Let's say you're a home cook and you are browsing through the reference section and you find a cookbook with a recipe that you really want to try. However, it's in a reference book and you cannot take reference books out of the library. Naturally, what you would do is make a copy of the information in that cookbook. You could make a photocopy. You could write it down. These days, you'd probably just take a picture of it with your cell phone, or, maybe scan it in if you wanted to. But one way or another, you could copy that information, take it out of the library, and bring it to your home or wherever you do your cooking. In that analogy, the book in the reference section of the library would be like the DNA. The book stays in the library. It doesn't circulate. It's not ever supposed to leave. That book contains information that you want for making something. So you copy the information into another format that can leave the library and carry that information to the kitchen where you actually do the cooking and assemble whatever dish it is that you wanted to cook. Okay. The thing that information is copied onto is a second type of nucleic acid called ribonucleic acid, or for short, RNA. RNA turns out to be that adapter. RNA is a molecule that copies information from DNA and takes it out to where proteins are actually made. Excuse me. RNA is like DNA in that it's a polymer of nucleotides, right? They're both nucleic acids. For your purposes, DNA and RNA are the only two nucleic acids we're going to talk about. The differences are, first of all, RNA is usually single-stranded. You can make double-stranded RNA in a lab, but in life, RNA is usually single, although sometimes you can have a single strand that will bond with itself and make a loop. But RNA does not naturally exist as a double helix. It's not two strands wrapped around each other. RNA, again, usually does not form a helix at all. RNA nucleotides contain a sugar called ribose, not the sugar called deoxyribose. And RNA uses one different base. Instead of the base thymine, T, it uses a base called uracil, abbreviated U. And then finally, the last major difference is that DNA does not normally leave a eukaryotic cell nucleus. And again, if it does, that nucleus is breaking down and the cell is about to die. But RNA is made in the nucleus, but shuttles out of the nucleus all the time. So you'll find RNA outside the nucleus of a healthy eukaryotic cell. Okay. On the left, that's a DNA nucleotide. This contains the sugar deoxyribose in green, and in yellow, the base thymine. There's an RNA nucleotide on the right, and you'll notice, first of all, there's an extra sugar right here. That OH right there is an oxygen with a hydrogen stuck to it. Sorry, that's an extra oxygen, not an extra sugar. Come on, Wagoner. Sorry, too much blood in my coffee stream this morning. And that makes this sugar a sugar called ribose, not the sugar with the missing oxygen right there, which is deoxyribose. So deoxyribose is missing that oxygen atom right there. Ribose does have that oxygen. And in place of thymine, there's a slightly different base that RNA uses called uracil. It uses A, T, and G, adenine, guanine, and thymine. No, sorry, adenine, cytosine, and guanine, A, C, and G. But instead of thymine, it substitutes a slightly different looking base called uracil. Okay, do I really want to get into this? Okay, let me take this for a few minutes more. And then we'll pick up with a review of this on Tuesday. Real quick for now, what happens in the nucleus is a length of DNA will unzip and RNA nucleotides are assembled. And I can now reveal that enzymes help this happen to make a strand of RNA that's complementary to one of the DNA sense strands. And this follows the same rules you should already know. On the DNA strand, A, T, G, and C bond with U, A, C, and G on the RNA strand. So if I have a strand of DNA that says T, T, A, G, C, T, if that's a DNA strand, the complementary RNA strand that forms here will be A, not T, but U, C, G, A. There we go. Once that RNA strand is completed, it peels off and the two strands of DNA come back together. And that RNA strand is called messenger RNA or mRNA. That's a diagram of what it looks like. This purple blob is an enzyme called RNA polymerase that does a lot of the work of assembly. And right here where RNA polymerase is active, the DNA double helix is unzipping and a strand of messenger RNA is being made using one of the DNA strands called the template strand as a pattern. The messenger RNA passes out of the nucleus and goes to a tiny structure called a ribosome. And messenger RNA slips into the groove on the ribosome. And in a way that I'm not going to get into yet, because we're going to pick this up on when we get back from break, the ribosome, and there's, by the way, there's many thousands of ribosomes in every cell. There's not just one. But the ribosome is where the messenger RNA's information is read and used to assemble amino acids in the right order. When we make a molecule of messenger RNA using DNA as a template, we call that transcription. And when the messenger RNA is used to assemble amino acids in the right order, we call that translation. This is the steep part of the mountain, but by the time we get to the peak, you'll be amazed at the stuff that we can see. And I'll be able to talk about things like, what viruses are, what does COVID actually do? What's the thinking behind the COVID vaccines? This and much more is what we'll be able to discuss once we get an understanding of what really happens in transcription and translation. Now, with only 10 minutes to go, I don't want to start on translation, so I'm actually going to let you goobers go a little early. Don't think I'm going to make a habit out of this, but I'm going to go ahead and stop the recording right there.